So my name is Imani Fennell, and I'm a junior chemistry major, and I am presenting this for my microbiology class. Today I will be presenting intestinal microbial metabolism of phosphatidylcholine and cardiovascular risk. This is presented by me, Imani Fennell, and this research was done in the Department of Cellular and Molecular Medicine at Lerner Research Institute, Cleveland Clinic in Cleveland, Ohio. So a brief overview of what I'm going to be talking about today is first the background information to get us acclimated to what we're going to be talking about. And then I'm going to speak about the research aim, then some methods and procedure as to how they carried out this experiment, the results of the experiment, and a conclusion. So what this paper was talking about is how recent studies in animals showed mechanistic link between intestinal microbial metabolism of the choline moiety and dietary phosphatidylcholine, or lectin, and coronary artery disease through the production of a proasterochloric metabolite, trimethylamine N oxide, otherwise known as TMAO. The phospholipid phosphatidylcholine is the major dietary source of choline, a semi-essential nutrient that is part of the B complex vitamin family. Choline has various metabolic roles, ranging from its essential involvement in lipid metabolism and cell membrane structure to its role as a precursor for the synthesis of the neurotransmitter acetylcholine. Choline can also serve as a source of methyl groups that are required for proper metabolism of certain amino acids, such as homocysteine and methionine. There is a growing awareness that intestinal microbial organisms, collectively termed microbiota, participate in the global metabolism of their host. Also, intestinal microbiota have previously been implicated in complex metabolic diseases such as obesity. However, the involvement of microbiota in the inception of atherosclerosis in humans has only recently been suggested. So in this picture, it shows how ingested phosphatidylcholine, the major dietary source of total choline, is acted on by intestinal lipases to form a variety of metabolic products, including the choline-containing nutrients glycerylphosphocholine, phosphocholine, and choline. Choline-containing nutrients that reach the cecum and large bowel may serve as fuel for intestinal microbiota, otherwise known as gut flora, producing trimethylamine, TMA, and then TMA is rapidly further oxidized to TMAO by hepatic flavin-containing monooxygenases, or FMOs. TMAO enhances the accumulation of cholesterol in macrophages, the accumulation of foam cells in artery walls, and atherosclerosis. All factors are associated with an increased risk of heart attack, stroke, and death. Choline can also be oxidized to betaine in both the liver and kidneys. Dietary betaine can serve as a substrate for bacteria to form TMA and presumably TMAO. So the research aim of this project was to investigate the relationship among intestinal microbiota-dependent metabolism of dietary phosphatidylcholine, TMAO levels, and adverse cardiovascular events in humans. Also, the relationships between oral intake of phosphatidylcholine and the involvement of the intestinal microbiota in the formation of TMAO in humans was investigated. And the relationship between fasting plasma levels of TMAO and long-term risk of incident major adverse cardiovascular events were examined. So 2 experiments were done to investigate these claims. So the first one included a phosphatidylcholine challenge in which 40 healthy adults with no chronic illness, no active infections, no antibiotics or probiotics within the past month underwent a dietary phosphatidylcholine challenge. And this challenge, for each participant, the baseline blood and spot urine samples were obtained after an overnight fast, which was 12 or more hours. At baseline, participants were given 2 large hard-boiled eggs, including yolk, which contained approximately 250 milligrams of total choline each, to be eaten within a 10-minute period, together with 250 milligrams of deuterium-labeled phosphatidylcholine, which is also called D9-phosphatidylcholine, as a tracer contained in a gelatin capsule. And serial venous blood sampling was performed at 1, 2, 3, 4, 6, and 8 hours after baseline, along with a 24-hour urine collection. 6 participants were given metronidazole, or 500 milligrams twice daily, plus ciprofloxacin, 500 milligrams once daily for 1 week. And this phosphatidylcholine challenge was done 3 times. And then as for analysis, this information was quantified with the plasma and urinary levels of TMAO and plasma choline and betaine levels by means of liquid chromatography and online tandem mass spectrometry after a phosphatidylcholine challenge in healthy participants before and after the suppression of intestinal microbiota with oral broad spectrum antibiotics. For the second experiment, they took 4,007 adults undergoing elective diagnostic cardiac catheterization, and the participants had no evidence of an acute coronary syndrome, and fasting blood samples were taken from all participants at time of cardiac catheterization. With this information that they received from these participants, they took plasma aliquots isolated from whole blood cells and EDTA tubes, maintained at 0 to 4 degrees Celsius until processing within 4 hours, and stored at negative 80 degrees Celsius. An aliquot from each 24-hour urine collection was spun to precipitate any potential cellular debris, and supernatant were stored at negative 80 degrees Celsius until analysis. TMAO, trimethylamine, choline, betaine, and their D9 isotopologues were quantified with the use of stable isotope dilution assay and high-performance liquid chromatography. Levels of TMAO in urine were adjusted for urinary dilution by analysis of the urine creatine level. And these participants participated in a 3-year follow-up, and that was marked. So, some results were that endogenous or unlabeled TMAO and choline and betaine were present in fasting plasma samples at baseline. Both TMAO and D9-TMAO were readily detected in plasma after the dietary phosphatidylcholine challenge at visit 1. Time-dependent increase in the levels of both TMAO and D9-TMAO were also observed postprandially. A strong correlation was observed between plasma levels of TMAO and the absolute urine TMAO level, and the urinary ratio of TMAO to creatine. Time-dependent increases in the plasma levels of both the natural isotopes and D9 tracer forms of choline and betaine also increased after the phosphatidylcholine challenge. The suppression of intestinal microbiota by the administration of oral broad-spectrum antibiotics for 1 week in 6 of the participants resulted in near-complete suppression of detectable TMAO and D9-TMAO after the phosphatidylcholine challenge during visit 2 in both plasma and urine. After the withdrawal of antibiotics and subsequent reappearance of the intestinal microbiota over a period of 1 month or longer, the phosphatidylcholine challenge during visit 3 again resulted in readily detectable and time-dependent changes in TMAO and D9-TMAO in plasma and urine. Major adverse cardiovascular event during the 3-year follow-up was measured or detected in the participants in the second study. The mean age of the participants was 63 years old, and 2/3 were men. The prevalence of cardiovascular risk factors were high. Participants who had major adverse cardiovascular events also had higher baseline levels of TMAO as compared with those who did not have cardiovascular events. As compared with participants in the lowest quartile of TMAO levels, those in the highest quartile had significant increased risk of event. And this table right here, this chart shows the different quartiles and the amount who had myocardial infarction or heart attack and stroke or death in the 3-year follow-up in the amount of years. So once again, those who, as compared with participants in the lowest quartile of TMAO levels, those in the highest quartile had a significantly increased risk of an event. So one of those conclusions from this chart especially is that increased levels of TMO remain significantly associated with an increased risk of death and non-fatal myocardial infarction or stroke. So with this table are the results of assays for TMAO and D9-TMAO, panel A and B, after the phosphatidylcholine challenge, before and after the administration of antibiotics with the intensity of stable isotope dilution assays measured by means of high-performance liquid chromatography. The arrows indicate retention time where authentic isotope-labeled TMAO standard dilute. Also shown are the plasma levels of TMAO and D9-TMAO at each visit. The plasma levels of TMAO were markedly suppressed after the administration of antibiotics and subsequently reappeared after the cessation of antibiotics, which indicates that the production of TMAO from dietary phosphatidylcholine is dependent on metabolism by the intestinal microbiota. This ability of oral broad-spectrum antibiotics to temporarily suppress the production of TMAO is a direct demonstration that intestinal microorganisms play an obligatory role in the production of TMAO from phosphatidylcholine in humans. Intestinal microbiota convert the choline moiety of dietary phosphatidylcholine into trimethylamine, which is subsequently converted into TMAO by hepatic flavin-containing monooxygenases. Although an association between infectious organisms and atherosclerosis had previously been postulated, studies looking at the role of antimicrobial therapy in preventing disease progressions have been disappointing. It is important to recognize that the choice of antimicrobial therapy in previous intervention trials was based largely on targeting postulated organisms rather than modulating the composition of intestinal microbiota or their metabolites. Furthermore, even if an antibiotic initially suppressed TMAO levels, the durability of that effect with long-term use remains unknown. So in conclusion, we have shown that intestinal microbes participate in phosphatidylcholine metabolism to form circulating and urinary TMAO. We also established a correlation between high plasma levels of TMAO and an increased risk of incident major adverse cardiovascular events that is independent of traditional risk factors. That is the end of my presentation. Thank you.